You know, inside every single one of your cells, there's this incredible blueprint, the code of life. But how on earth do you read a story that's billions of letters long? Well, today we're going to decode the decoders. I want you to just try and imagine reading a book with 3 billion letters and no spaces, no punctuation. That's the huge challenge we're talking about when we sequence a human genome. I mean, the scale is just, it's mind-boggling. So, right, how do scientists even begin to tackle this? How do they take this invisible molecule and turn it into something we can actually read on a computer? We're going to dive into 3 really clever methods that do just that, and each one has its own totally unique way of getting the job done. You know, this whole quest to read DNA has been a real sprint. I mean, Watson and Crick showed us the double helix back in '53, right? But it took all the way until 1979 to get the first solid method, which we call Sanger sequencing. The real game changer, though, that happened in 2005 with next-generation sequencing. That's when we went from a slow crawl to, like, light speed. All right, first up, let's talk about the OG, the original champion, Sanger sequencing. The best way to think about this one is to imagine a tiny, microscopic race. And the runners? They're all different-sized pieces of DNA. So the big idea here is actually super simple. It's all about size separation. See, if you can make a bunch of copies of your DNA that stop at every single letter, and then sort all those copies by how long they are, you can basically piece the whole sequence back together one letter at a time. Okay, so how in the world do you make all these different-sized pieces? Well, there's this really clever trick. It involves a special ingredient that acts like a chemical stop sign. Scientists have a fancy name for it, a DDNTP, but all you need to know is that when this thing gets added to a growing DNA chain, the copying process just stops cold. Do that for every possible letter, and what you get is this perfect ladder of DNA fragments, where each rung is just one letter longer than the last. And now the race is on. All those fragments get loaded into this gel, which is kind of like a dense molecular obstacle course. Then an electric current pulls them through. And just like any race, the little guys, the shorter fragments, can zip through that mesh way faster than the big, clumsy long ones. And here we are at the finish line. As each piece of DNA, from shortest to longest, crosses the line, a laser zaps it, and it gives off this little pop of colored light. Maybe green means A, blue means C, you get the idea. By just recording that sequence of colors flashing by, we can read the DNA sequence. It's simple, it's really elegant, but yeah, it's pretty slow. So Sanger was clever, but it was slow. The next generation of technology, it took a totally different path. So let's leave the racetrack and enter the forest of light. This is where we go from a single race to a massive parallel universe of races all happening at once. I mean, the difference in scale here is just, it's staggering. Think about it. While Sanger is carefully reading one piece of DNA, Illumina is reading billions of them at the exact same time. The key idea here is what we call massive parallelism. So forget that single race lane. Instead, I want you to picture a microscopic stadium with billions of different reactions all happening at the same time on a glass slide that's literally smaller than your thumb. And check out how this all gets started. It's pretty cool. A single DNA molecule basically sticks to the slide, it bends over into an arch, and then it gets copied. This happens again and again exponentially, kind of like one seed sprouting into a whole dense forest of thousands of identical DNA strands. We call that a cluster. And then comes the light show. The machine washes in one letter at a time, and each one has its own unique color. Every single cluster on that slide grabs the new letter and lights up. Flash! A photo gets taken. Then, whoosh! Everything gets washed away, and the whole cycle starts over for the next letter. The entire slide just twinkles like a massive field of Christmas lights. And the numbers here are just, they're hard to even wrap your head around. A single run on an Illumina machine can produce over 2 billion separate DNA reads. That's not just reading one book anymore. That's like reading an entire library simultaneously. Okay, but what if you could just get rid of all of that? No copying, no light shows, none of it. Well, that brings us to our third method, which feels like something straight out of science fiction, the nanopore gatekeeper. So with this one, the whole strategy just gets turned on its head. We're talking about physical detection. No more chemical tricks, no more light shows. This method literally feels the DNA strand as it's pulled through a microscopic hole. Okay, here's the breakdown. You've got this tiny protein gatekeeper, a nanopore, stuck in a membrane. A motor protein grabs a long single strand of DNA and basically forces it through that tiny hole. And as each letter, A, T, C, or G, gets squeezed through, it messes with an electrical current in its own unique way. The machine reads that electrical disruption, that sort of thump, thump in the signal, and knows exactly which letter just went through. It's just such a radically direct way of doing things. You don't need to make copies of the DNA to create a forest. You don't need a whole series of washes and photos. It's just one long, continuous strand of DNA being read, letter by letter, in real time as it zips through the pore. And the advantages here are pretty incredible. First, it can read really long pieces of DNA, which is a lifesaver for putting together the tricky parts of a genome. Plus, the data streams off the machine as it's happening. It's real time. And get this. Some of these sequencers are so tiny, you can literally plug them into a laptop and do sequencing out in the middle of a field. So, there you have it. We've got the slow and steady race, the massive forest of light, and the futuristic gatekeeper. So, which one's the best? Well, the real answer is, it totally depends on the question you're trying to answer. You can see here they each have their own superpowers. Sanger, it's still on the gold standard if you just want to double-check a single specific gene, something up to about 1,000 letters long. Illumina is the absolute king of counting things, like finding out which genes are switched on, because it gives you billions and billions of short reads. And Nanopore, it's the champion for reading super long, unbroken stretches of DNA, which is exactly what you need if you're trying to build a brand new genome from scratch. So when it comes right down to it, here's the main takeaway. There's no single best method. What's happened is that this explosion in sequencing tech has given scientists this amazing, diverse toolkit. And the real skill is knowing exactly which tool to pull out of the box to answer your specific scientific question. I mean, think about it. We've gone from a single race to a forest of light to a molecular gatekeeper. Our ability to read the code of life is it's breathtaking. And it leaves us with one last really big question. Now that we can read pretty much any biological code we want, what stories are we going to write next?